Bismillahirrahmanirrahim. As others have already mentioned, the topic today is unlocking the power of immunotherapy for cancer. For this, it is important to understand what cancer is. Cancer is an abnormal cell cluster which expands and really grows fast, uncontrolled. Under normal circumstances, immune cells will attack the cancer very early and stop the cancer cells to grow. But cancers, as a lot of doctors are in the room, know a way to evade the immune system. To understand how the evasion works, it is important to understand what is in the environment around the cancer, so the so-called tumor microenvironment. There are not just tumor cells in this cancer cells. You will have various immune cells, including T cells, myeloid cells, regulatory T cells, dendritic cells, macrophages. These are all immune cells which have a good interaction with each other. At the same time, there are a lot of cancer cells which interact with fibroblasts. Most importantly, T cells play a very important part in fighting the cancers. T cells are the so-called warrior of the immune system, which are mainly involved in fighting cancer, but also infections. The CD8 T cells, which are primarily fighting the cancer, they need a certain activation to work against the cancer cells. This activation or the interaction is required through the T cell receptor, shortened here with TCR. And this TCR activation needs a MHC complex. MHC can also be called HLA, because later on we are going to talk about HLA. And on this MHC complex, the antigen, which is mainly expressed by tumor cells, is being presented to the T cells so that the T cells can get activated, expand, and then kill the cancer cell. Unfortunately, it's not that simple. Cancer cells have ways to evade this T cell activated killing. There are various immune suppressing molecules expressed by tumors, but also by the cells in the tumor microenvironment. Some famous ones are PD-L1, CTLA4, but also TGF-beta. The expression of these molecules results in the so-called cold tumor, that you have a tumor with less T cells inside the tumor microenvironment rather in the surroundings. And the aim is to actually have a hot tumor to kill the cancer. At the same time, what tumors also like to do is they like to recruit regulatory T cells into the tumor microenvironment to actually dampen the immune response. Regulatory T cells are actually used to downregulate potentially autoimmunity. Here they are kind of misused by the cancer. If we look some years ago, there was a Nobel Prize being awarded for the immune checkpoint inhibition. Here, specifically, antibodies were developed to inhibit PD-L1, PD-1, or CTLA4. This resulted in the activation of the exhausted T cells, and you were seeing, or you were starting to see of cells which were not able to kill the cancer. They started killing the cancer cells again. This was a very big breakthrough in the immunotherapy field. Unfortunately, there are cancer cells which still have ways to evade that. We are coming back to this figure. Means, unfortunately, it's not just very simple to get exhausted T cells back fit. You will have various other molecules. TGF-beta, for example, was not mentioned. There's no way of downregulating TGF-beta that easily. But also at the same time, you have other regulatory T cells in the soup, which inhibit the formation of a hot tumor. The question is, how do we get to the point that we can actually treat patients more successfully in to beat the tumor? And there are, in the current market or in the current situation, we have various different ways. And most of these modalities which are now being developed is very much a personalized medicine. Here we are trying to learn from patients and identify targets to treat the patients. There are three different things marked here. One is the cancer vaccines, where we are sequencing for new epitopes or new antigens in the tumor to develop and train the immune system to identify these new epitopes, and then develop the T cells to fight the cancer. At the same time, we have chimeric antigen receptor and T cell receptor therapy. Both rely on engineering of T cells. In one case, we are developing a chimeric antigen receptor to mark a specific molecule which is being expressed on the surface of tumor cells. This therapy works very nicely in liquid tumors where we have B-cell lymphomas and others, where we can actually deplete the whole population. Unfortunately, it has its limitation on solid tumors. T-cell receptor therapy, on the other hand, that's the one what I'm working on. It relies more on a natural expression of the antigen, and you are relying on a more natural T-cell receptor. Coming back to the initial how T cells are being activated, they rely on the T cell receptor, which is being expressed on the surface of the T cells. They recognize on the MHC complex or HLA of the tumor cells the antigen which is being expressed in the tumor, and then start producing the cytokines, the chemokines, and other molecules to kill the tumor. As I said, a tumor or a cancer is very smart. It can downregulate certain antigens. It can downregulate certain HLA molecules, and it can produce various hostile molecules like TGF-beta, PD-1, etc., resulting in downregulation of the therapy. To address this, we at IITs can mainly work on finding a way of how to improve a potential cell therapy where we are planning to target these different problematics. First of all, we have to identify the patient. We have to understand what is the tumor profile of the patient, what antigens are being expressed, what is the HLA type of the patient. And then we go into the immunobank which we have developed, where we are taking the different TCR-Ts, the so-called engineered T cells, and then we are attacking the cancer from different fronts. We have an antigen targeting a specific HLA, then we have a different antigen on another HLA, so that if the cancer tries to evade, we have a way of fighting it. So that's why we are trying to optimize and to use a multiplexed approach to beat the cancer. And inshallah, hopefully, we will see soon some good results in that. I wanted to conclude with a verse from the Holy Quran. All therapies are good, but at the end, we have to rely on, as it's mentioned in the Quran, on Allah Ta'ala. As it's written here, and when I'm ill, it is He who restores me to health. And all therapies are good and well, but at the end, it's all in Allah Ta'ala's hand. Jazakallah.